Okay, class 12, we'll continue with the second lesson of your NCERT textbook, the sexual reproduction in flowering plants. This is the second class for this chapter. Quick revision. We did structure of flower in previous session. Floral parts are broadly divided into two worlds, the essential and the non-essential. The essential include primary sex organs, that is the male reproductive part, and the female reproductive parts. The non-essentials include accessory organs, the petals, sepals, and also there is pedicel or the floral stalk and the thalamus or the receptacle. Thalamus or receptacle is the flattened part of floral stalk which bears the floral whorls. In chapter 1, we studied that every cycle of sexual reproduction is made of three events: pre-fertilization, fertilization and post-fertilization. Same happens in plants. So in plants, pre-fertilization events comprise of formation of male and female gametes. Now formation of male gamete is known as microsporogenesis and formation of female gametes is known as megasporogenesis. Microsporogenesis is usually formation of microspores, which is the pollen grain. This pollen grain contains the male gamete. Similarly, megasporogenesis is actually formation of megaspore, which is ovule, and this ovule contains the female gamete. Now I want you to look very carefully at this diagram, draw this diagram, and never ever forget these terms and the sequence in which these are formed. So in this session, we'll try to cover the first part of pre-fertilization events, that is structure of male reproductive part, stamen, and the process of microsporogenesis along with the structure of microsporangium. In angiosperms, the main plant body is a sporophyte. So the flowering plants that you see around you are sporophytes which reproduce through spores. So in angiosperms, there are two types of spores. The male spore is known as microspore and the female spore is known as megaspore. Microspores are borne on special leaves known as microsporophylls. These special floral leaves are stamens. Stamen is made of two parts, a long slender part known as filament, and at the tip of the filament is the anther. So anther is at the distal or farther end of the filament and the proximal end of filament is attached to the thalamus or receptacle. Now anther is the fertile parts, which means it will bear the pollen grains. Now you can see anthers are bilobed structures. Bilobed means there are two lobes which are separated by a groove. Now each of these lobes are further divided into two theca or bags. So each of the anther lobe is dithecous. A longitudinal groove runs in between the two lobes of anther. This is connected at the end by a tissue known as connective tissue. This connective tissue is sterile and it is made up of parenchyma. Now each lobe of anther has two pollen sacs. These are the microsporangium. The microsporangium is a cylindrical bag, as you can see, and when we study the transverse section, it appears circular. So this is the standard structure of stamen, but if you take different flowers, you will see many variations. Number of anther, their attachment, fusion, etc. are very different in different species. Now here is a transverse section of anther. You can see the two lobes, one on the left side and the other on the right side. Now each lobe has two round structures marked as sporogenous tissues. So 1, 2, 3 and 4 sporogenous tissue. So anther is generally said to be tetrasporangiate, or 4 sporangiums are present in this. We'll come to that later. Now here this sporogenous tissue develops into microspore mother cell. We also call it pollen mother cells, or in short PMC. These will later form the spores or pollen grains. Now you can see outside there are 4 layers of cells: epidermis, endothelium, and there are 1 to 3 middle layers. So 4 types of layers are present: epidermis, endothelium, middle layers, and inside is the tapetum. Now except for tapetum, the remaining wall layers, that is epidermis, endothelium and middle layers, provide protection. They also help in dehiscence or release of pollen from anther. What does tapetum do? Tapetum provides nutrition. Cells of tapetum are larger compared to the outside cells, and they are dense cytoplasm. So they are loaded with nutrients and may also contain more than one nucleus. So tapetum is supposed to provide nutrition to the developing microspores inside. Besides these, tapetum performs one more very important function. It secretes lipid-rich granules known as Eubish granules. These Eubish granules form exine, or the hard outer cover of pollen grain. This exine keeps the pollen safe. So two functions of tapetum: nutrition and secretion of Eubish granules. Now what you see here is TS of the anther. You can see the filament in the center with anther lobes on 4 sides. Tapetum is visible in pink color with pollen in the center. Now microsporogenesis, we understand, is the process of formation of microspores. Now the story of microspores starts with the mass of sporogenous tissues. You recall where they were? In the anther lobes. Some of these sporogenous tissues develop into pollen mother cell, which will form microspores or pollen grain. Now sporogenous tissue and hence pollen mother cell are part of the parent sporophyte, so they are diploid, that is 2n. But the spore will have to be haploid because it has to give rise to the gametophyte generation. Remember, alteration of generation? So PMC will have to undergo meiosis or reduction division. Now in meiosis, one parent cell forms 4 daughter cells. So each pollen mother cell gives rise to pollen tetrads, as you can see. Later these tetrads separate and thus 4 pollen grains are formed from each PMC. Thousands of such pollen grains are simultaneously produced in a pollen sac. When the anther dehisces, all these are simultaneously released. Now let's study the structure of pollen grains. Once again, pollen grains are the male gametophyte. Before we go into detailed structure, let's see a few pollen grain images using the amazing technique of scanning electron micrography. Now what is striking about these pollen are the unique surface features. The colors, designs, each of them are unique. Here is the structure of a microspore or a young pollen. It is spherical with 2 wall layers, the outer exine and the inner intine. Now, exine is special because it is made of a substance known as sporopollenin, which is highly resistant fatty substance. It can resist high temperature and even acids. In fact, We don't know yet of any enzyme that can degrade sporopollenin. How is this sporopollenin formed? Do you recall? We just covered it. Now, why is sporopollenin important? You know, pollen grain is released outside. It may or may not land on correct stigma. It can be eaten by predators or may just be scattered. Either way, pollen grain survives because of the resistant nature of sporopollenin. Fossils of pollen are also remain preserved because of this. Now you can see certain depressions on the surface of exines. These depressions are known as germ pores. These are gaps left in the exine to allow the pollen tube to come out. The number and position of germ pore along with the exine structure is unique to a species, and so we use this feature in taxonomy. The study of pollen is therefore very important and is known as pollinology. Intine, on the other hand, is made of cellulose and pectin. Now, at younger stage, pollen is made of one cell with one nucleus. But this being microspore, it will divide to form the male gametophyte and then become the mature pollen grain. So, this microspore starts to develop inside the microsporangium itself, and this type of development is known as precocious development. So young microspore has one cell, one nucleus. This cell divides by mitosis after finding an asymmetric spindle. This means that this mitotic division is unequal. A large vegetative or tube cell is formed and a small generative cell is formed. Now this generative cell is spindle shaped with a nucleus which is spindle shaped and less cytoplasm. The vegetative cell has dense cytoplasm which is rich in food reserve. Its nucleus is also large but irregular. Now this is the two-cell stage of pollen grain which is released for pollination in 60% of the plants. In these plants, the generative cell divides further but only after the pollen grain lands on stigma. This generative cell after dividing will form two male gametes. So in 60% of the plants, the pollen grain is released at this two-cell stage. But in some, the generative cell also divides while it is with the parent plant, that is inside the microsporangium. This generative cell forms two male gametes, and then we have the three-cell stage, the two male gametes and the generative nucleus or tube nucleus. So this is the three-cell stage, and in some cases the pollen is released at this three-cell stage. Now here is the compiled list for comparison between the two cells of pollen grain. With this we come to the end of the process of microgenesis. But before we end, few words about the significance of pollens. First, the bad news. Pollens lead to allergies, respiratory problems and bronchial afflictions. You recognize this plant? This is Parthenium or carrot grass. It came to India accidentally as contaminant with wheat, and now not only it is difficult to control, its pollens are a nuisance during blossoming season, leading to allergies. For some people, the pollen season is very difficult. Okay, now the good news. We just saw that pollens are rich source of nutrients. So they are marketed as tablets and syrups to be used as food supplements. They are used for enhancement of stamina of athletes and racehorses. Finally, the viability or the life of pollen. It is dependent on the species and on the temperature and humidity of the area in which the pollen is present. Now we can see rice and wheat, the pollen viability is only for 30 minutes, but for the plants of family Rosaceae, Solanaceae and Leguminosae, the viability is for 3 to 4 months. But the pollen can be stored in pollen banks at minus 196 degrees Celsius using liquid nitrogen, like we make sperm banks or seed banks for later use. Now these pollen which are stored in pollen banks can be used for breeding programs. Finally, the question time. Try to solve these after you have understood the content. Try to practice the diagrams. We'll continue this again. Bye-bye.